My name is Bill Mahoney, and I direct the M3D PhD program, and welcome to Path Presents today. I'd like to welcome an alumni from our program, Nate Basisty, who is here both as an undergrad and as a grad. And I'm going to hand it over actually to Peter Rabinovich to give a more personal intro. I just want to remind people, normal Zoom rules, mute yourself during the presentation. When we get to the end, I'll look for questions in the chat. And if there aren't any there, you can raise your hand and I will call on you to ask your question. But let's use the chat as the talk goes on, and then we will, I'll moderate that at the end. With that, I will hand it off to Peter for a mentor's intro of a mentee. Take it away, Peter.
Thank you, Bill. This is a real pleasure to introduce Nate. And we'll talk about Nate as an example of a great trajectory, and I'll mention that again at the end. But Nate came to us, as Bill mentioned, from UW as an undergrad in biochemistry, entered here in 2010 to our training program, and was also accepted into our aging training program a couple years after that. His thesis work was on proteome turnover in mouse aging, using stable isotope mass spectrometry, and was co-mentored by Mike McCoss, who has joined us also for this presentation. So Nate was able to publish 7 papers, 2 of them first authored and 2 of them co-first authored during his time. He was also supported by a Glenn A. Farr Scholarship for research in the biology of aging. Nate first worked on the proteomic effects of caloric restriction and rapamycin in mouse hearts and livers, and as part of that work, he helped the McCoss lab develop a metabolic labeling strategy for measuring protein turnover rates using heavy leucine in feeding of mice. And that really was a breakthrough at the time for looking at proteome-wide turnover rates. Nate also studied polyubiquitinated proteins in aging, and there also applied the protein turnover measurements, work that was published in the journal Aging and received the editor's choice when it was published. Nate was also the lead author on a study on proteomics of MCAT mice, and the MCAT mice proteomics was especially interesting as an example of antagonistic pleiotropy. In other words, the proteome showed that while the proteome of old mice that were MCAT returned closer to that of young mice, in fact, the young mice that were MCAT had the reverse. They had a proteome that had several features, many features, that were more characteristic of aging. This was a real surprise for us, and we had to ask Nate to redo that work several times because it was almost unbelievable. It was in fact quite true. It was a great example of antagonistic pleiotropy, and when published, it was recognized by the receipt of the Joseph Pignolo Senior Award in Aging Research, and it was a Aging Cell Best Paper Prize in 2016. Nate went on in 2015 to be a postdoctoral fellow at the Buck Institute for Research in Aging in Novato. His primary advisor was Birgit Schilling. He was assisted by Judith Campisi, and he published 30 papers, 7 of them first authored, so very, very productive. He was also supported by a Glenn Foundation Research Training Fellowship. Within the past week, Nate has decided what he's going to do for the rest of his life, or at least the next part of it. He's going to begin as a tenure-track investigator in the Translational Gerontology branch of the National Institute of Aging, running the Translational Geroproteomics Core, or unit. So that's really, it's a great end to a trajectory. It's really a great example. I think our trainees will enjoy talking to Nate, because Nate, I think he himself will admit that he was a diamond in the rough. There were some sharp edges when he started the program, and I take a lot of pride in the program and the mentors here in helping to polish that diamond. And now you can see from the trajectory that Nate has really just continued on an upward path towards expertise in geroproteomics, culminating in this tenure track position. So Nate, we're real proud of you. We're happy to welcome you back to present your more recent work. And thank you for joining us. It's great to see you.
Oh, thank you so much, Peter. That's probably the best introduction I've ever gotten. Thanks. And, you know, I'm really, really happy to be here because, as Peter said, you know, this program and this department is where I got my start even doing proteomics. I mean, I got my start doing proteomics here, and I got my start doing geroscience here. And, you know, as Peter said, it wasn't an easy journey, but it was, you know, ended up working very well. And, you know, I'm really happy that I did it, and I'm just really happy to be back. And now tell you about some of the work that I've been doing. So yeah, today I want to talk to you about some of the work I've been doing, a little bit from UW, but a lot of the work I've been doing now as a postdoc. I'm a postdoc now at the Buck Institute, and here's an aerial shot of the Buck. And you can see I really encourage if things ever return back to normal, that you visit this campus, because I think it's at least worth visiting the Buck, especially, not only for the aging research, but I think it's also just a beautiful campus. But anyways, so I don't think I need to describe to this audience why it's important to do geroscience, because I think there's a very strong geroscience component in this department. So I'm only going to spend this one slide doing it. But aging is the greatest risk factors for most chronic diseases, right? And as we age, our risk of all these different diseases, including the big killers like heart disease, cancer, and Alzheimer's disease, goes up. And when we reach middle age, our risk of all of these things goes up, exponentially. And death follows a similar trajectory. And and I'm not saying this to depress you. In fact, actually, in the geroscience field, there's an upshot to this, which I'll get to in a second. And one of the basic hypotheses of geroscience is that this isn't a coincidence, that there are basic aging processes that are driving the susceptibility to multiple age-related diseases simultaneously. And here I'm highlighting two of these, which are loss of proteostasis and senescence, but of course because these are the ones I'm going to focus on today, but of course there are many others. There are, DNA damage, mitochondrial dysfunction, inflammation, et cetera, et cetera. And this is actually in a way good news, because if this is true, then we should presumably be able to intervene in these aging processes to treat multiple age-related diseases simultaneously. So we can make this blue line look more like this green line. And this is not only like a huge public health benefit, but also a huge economic benefit, right? Because if you could treat multiple age-related diseases this way, that would be much cheaper than trying to cure all of these complex diseases late in life, right? So there's a lot of benefits to taking this approach. And the other good news is that we've shown that this approach works, at least in model organisms. So for example, we have interventions like dietary restriction, which target these pathways, which we know will extend lifespan in a variety of organisms, from yeast to invertebrates to mice. And I think dietary restriction would work in humans as well. Rapamycin and also senolytics. So these are drugs that selectively kill senescent cells. So it's really important, I think, to use the tools we have like proteomics to characterize these processes at the molecular level, because this helps us to identify targets for translation into humans, but also allows us to quantify things like the rate of aging and use these as biomarkers to aid us in translation of therapies. So today, I'd like to cover a few different topics, and I'd like to start with some work that really started when I was here at UW working on protein turnover and its role in aging and longevity, but now is something that I've been revisiting in recent years. And then the main focus of my postdoctoral work, which was on cellular senescence, and then some ongoing work I'd like to do as I open my own lab. So before I get into this, I just want to acknowledge that a lot of this protein turnover work, which I'm going to try to summarize very quickly, a lot of this was done in collaboration with other people in Peter's lab, including Ann Chow, Daofu Dai, and Pabalu, and who have all now moved on to really nice positions actually all over the US. So Peter, you've had a lot of successful people coming out of your lab actually. And really, we really collaborated on a lot of this. I mean, this was a whole, series of different papers, and we collaborated on a lot of these studies together. So I just want to acknowledge that they were closely involved in anything that I'm going to show you now. But really, I just want to focus a little bit on a study that was actually Dao Fu took the lead on, because this is one of the data sets that I'm now working on to develop tools for protein turnover. And this is a study that's focused on protein turnover in cardiac aging and the effect of calorie restriction and rapamycin. And so at the time when we were doing this study, we were really interested in the Mtor pathway. So the Mtor pathway is a really well-known and probably the most studied pathway in aging. And it's a conserved regulator of aging from yeast to mice and all the way up through mammals. And mTOR is essentially a proteostasis pathway as well. So what we know about mTORC1 is this is a nutrient sensing complex. So if nutrients are high in the cell, it senses levels of glucose, amino acids, and insulin and other cellular cues to then fine tune downstream processes like proteostasis, so protein synthesis and autophagy. So when there's a nutrient excess, mTORC1 is activated. And this tells the cell that, okay, we have the nutrients to grow, right? So let's now, in line with that, let's increase protein synthesis and let's decrease autophagy. But now if you inhibit the mTORC1 complex, either by reducing these incoming signals with calorie restriction or pharmacologically with rapamycin, now you're telling the cells, okay, we don't have a lot of resources, let's go into conservation and maintenance mode. So in turn, the cell is going to turn down protein synthesis because it can't afford to synthesize proteins, right? And upregulate autophagy because it wants to degrade the proteins that it has into amino acid constituents, which it can then prioritize for the proteins that it needs, right? And so all of what I'm just telling you now is something that's been long known about the mTOR pathway. But at the time when we were doing this study, one of the things that's less known is what happens when you inhibit this complex for a longer period. So in the short term, yes, this is exactly what happens. Protein synthesis goes down and autophagy goes up. But most aging studies are done over weeks or even longer, right, of these kind of treatments. So the question is, you can't have reduced protein synthesis and increased protein degradation for weeks or longer, because eventually you'll just run out of proteins, right? So the question, the proteome has to reach a new steady state. So the question is, what is that steady state? How do calorie restriction and rapamycin alter global protein turnover, and which proteins, importantly, are the most affected? And so to answer questions like these, we turn to metabolic labeling in combination with mass spectrometry. So what we did is we took mice who were treated, who were either treated with calorie restriction, which is a 40% reduction in calories without malnutrition, rapamycin, which is microencapsulated 14 parts per million in the chow, or untreated mice, both young and old, treated for 10 weeks on these respective treatments, after which we introduced a heavy leucine into their diet. So now at this point, the mice are now over what we call this metabolic labeling period. And so what this does is now the mice are eating this heavy leucine, and as heavy leucine gets into their cells, new proteins that they synthesize, right, will be incorporating heavy leucine. And so downstream, when we do mass spec analysis, we can actually look at this incorporation to tell us, to figure out what is the, how many new proteins are we making. And so during this 17-day labeling period, we collect tissues from mice from all the groups at 4 different time points. And in this case, I'm going to be telling you about the heart, but we've done this now in multiple different tissues and models, and do mass spec analysis. And then we do the data analysis part. So the data analysis, in our case, we used an in-house software, and this was actually software developed in Mike McCoss's lab with Nick Schulman and Ed He. And what this software does is it allows us to now look at our mass spec data and look at peptides with multiple leucines, and then look at the, do what's called a mass isotopolog analysis, and uses that to accurately estimate the percentage of protein that's newly synthesized in our samples. I then took this data and did regressions of newly synthesized protein over time using in-house R scripts, and then also did statistical analysis of, how are these protein half-lives changing between, say, young and old mice, or old mice and old treated mice, to find significantly changing proteins. And then we would do functional, or look for potential functional changes using ingenuity pathway analysis. And essentially what we found when we did this study is that we were able to measure the half-lives of hundreds of proteins in the mouse heart. And what we found was that in old mice that were treated with either calorie restriction or rapamycin, so both of our interventions, what we saw across the entire proteome was in general that proteins were longer lived. But in particular, we saw that the strongest effects were seen in proteins involved in mitochondrial metabolism and TCA cycle. And at the time, I think this is really interesting because this pointed us in the direction of maybe these interventions are changing mitochondrial metabolism. And this was confirmed using targeted metabolic profiling. So we saw that, in fact, old mice treated with rapamycin, they have improved mitochondrial metabolism. And at the time, and this was really interesting too, because we saw this was correlated with improved cardiac function. So essentially, these mice had rejuvenated heart function. And I'd like to highlight this example of a turnover study because I think it really highlights that you can use proteomics and do proteomic measurements of turnover, and this can actually point you in the direction of a mechanism that you see with an age-related improvement. So this is, I think this is a really good example of that, and I think it highlights the importance of looking at protein turnover. But this was just the first of a series of studies that we did on protein turnover, and if I summarized what we found in all of the studies that came out of this research in the Rabinovich lab and just looked at one common denominator between everything, one really interesting observation that we made was that in all of the different longevity models we looked at, which include calorie restriction, rapamycin treatment, and mitochondrial targeted catalase. So these MCAT mice, I think many of you are familiar with these. That were made in Peter's lab. These are transgenic mice that overexpress this enzyme catalase targeted to the mitochondria. That in all of these models and all of the tissues that we looked at, what we saw was that mice that were given these treatments had longer proteome half-life. So it seems that in all these different interventions, longer protein half-life is correlated with extended life, right? So, I mean, so when we did these studies, I thought this was a really interesting and begin to raise the question, are longer protein half-lives a requirement for longer life? And while we were doing these studies, other labs had verified some of our findings. So the Hellerstein lab at UC Berkeley, they also published a study that looked in calorie restriction and rapamycin, but in addition, and confirmed our findings, but in addition, they looked at the Snell dwarf mice. So this is another transgenic mouse model that's long-lived. And again, they found that in this mouse, there were longer proteome half-lives. And in fact, in their study, they used 2 different doses of rapamycin and showed kind of a dose-dependent increase in protein half-lives, which is also associated with dose-dependent increases in lifespan. So this was really interesting. And even more recently, people began to look at long-lived other animal models. So more recently, the naked mole rat. So Vera Gorbanova and Sina Gamagami at University of Rochester had more recently shown that the proteome half-lives in the long-lived naked mole rat are longer than their mus musculus counterparts. And then Shelley Buffenstein, who's the other naked mole rat person that you probably know, is who's in Calico, the Google spin-off company who works on aging. She dropped a paper in bioRxiv showing the same thing, that naked mole rats, which are very long-lived compared to mice, have longer protein half-lives. And most recently, I think what's really interesting is that Sina Gamagami put out another study where he did a comparative analysis of mammals with a diverse array of maximum lifespans. And what he showed is that in this study, he showed that there's a beautiful correlation between the turnover of the proteome, almost linear correlation of the turnover of the proteome and the maximum lifespan of these animals, including, and this study included the longest lived mammal, which is the bowhead whale. And so I think all of this really together shows that proteome half-life seemed to be a really important factor in longevity. And it really begs the question, are longer protein half-lives a requirement for longer life? And I think there's a lot of interesting studies that are yet to be done to get at this question. And I think really what this shows is that these kind of studies provide really important insights into the biology of aging. And I think it'll be and I think there's demand to do more of these kind of studies. But the problem is that these studies are also very difficult to do. So these studies can essentially be broken down into 2 components. First is the metabolic labeling phase. So this is, for example, when you introduce heavy leucine into your mice, right? But then there's the data analysis phase, and this is really where the bottleneck lies for most labs. And the problem is that there's only a handful of labs that have the tools in place to properly calculate protein turnover from this kind of data. And of the labs that do, including our own, they're usually developing their own in-house pipeline, so it's not as accessible to other labs. So one of the things I've been really focused on for the last few years is to now take our in-house pipeline, to rewrite it and repackage it into in R, and create this tool called Turnover. And what this now enables us to do is create this an external tool for a really well known and recognized and used open source proteomics platform called Skyline. And so Skyline is a really nice proteomics platform. If you ask a proteomics person, they'll know what it is. This is a software tool developed by the Makas lab here at UW. And so we've been collaborating with Mike and the Skyline core development team, including the lead developer, Brendan McLean, and Nick Schulman, who actually wrote the original topograph software, to now to incorporate the capability of turnover analysis in Skyline. So I just want to point out a few features, important features that are now added to the Skyline software that enable us to do this kind of analysis. So now what's really cool is that in the Skyline software, they've already incorporated these really these key features. One which is called Permute isotope modification. So in Skyline, when you've loaded all your this experiment, experiment into Skyline, now you can use this feature. And what this does, that's really important for our analysis, is it takes these peptides that contain multiple leucines and it populates your Skyline document with all the different permutations or the different isotopologues of this peptide. And so what I mean by that is that we need to know how much signal is coming from the version of this peptide with 3 light leucines, the version of this peptide that has all heavy leucines, but also the version that has just 1 heavy leucine or has just 2 heavy leucines, right? We need to know all these isotopolog contributions in order to do turnover analysis. So this is something you can now do in Skyline. And so once you've permuted these modifications, you can open up our tool, type in a few settings and push go. Turnover performs the isotopolog analysis. It performs the regressions of the proteins and calculates half-lives. And something that's really unique to our tool now is that it also does statistical analysis. So it'll tell you which proteins are significantly changed between your treatment groups, and then spit out summary reports and comprehensive reports of all the protein changes. So as proof of principle, what I've now done is taken all of the data from that experiment I just showed you in calorie restriction and put it through our tool. And I just want to show that some of the cool information you can get out of our tool. So we were able to reproduce the results from the previous study we'd done with calorie restriction in mouse hearts. And here on the left, what I'm showing you is an example of a global summary plot of what you can see that's outputted by the turnover tool. And what this is showing is that you can nicely see both in calorie restricted and control mice that newly synthesized proteins are being incorporated over time, and that this incorporation is significantly slower in calorie restricted mice, right? And we can also look at all the protein by protein changes. So here you can see 4 proteins that had significantly slower incorporation of new proteins in calorie restricted mice versus wild type mice. So here in the blue lines, you can see calorie restricted mice versus wild type mice. So we've shown nicely that we can actually recapitulate changes that we've seen before using turnover tools with this. And so now, as we launch this tool, something I'm really interested in doing is now to take it and to ask, answer some outstanding questions about, in the field of protein turnover and aging. So, some of the really interesting questions to me now are which proteins are turning over in specific age-related diseases. For example, sarcopenia, where you might see, muscle atrophy, right? Or how do PTMs affect protein turnover? And we've done a little work on this. So, ubiquitination, oxidation, acylation, do these affect rates of protein turnover? I'd really like to get at this question of, is longer half-life a biomarker for whether an intervention will extend lifespan? And so, this is a tool that we can use to examine that. And then finally, how do insoluble proteins change during aging? So everything I've shown you up until now has been insoluble proteins. But with age, we know that insoluble proteins are aggregating and you know, in insoluble proteins, you might expect things to go the opposite direction. You want to see those actually turned over more in those aggregates removed in an aged organism, and that you would expect that to be more of a marker of health. But now the first question the question I'm first going to apply this tool now to is the first question here, which is which proteins turn over during sarcopenia? And the reason for this is because we already have a nice collaboration where we can actually do this. So we're working, this is everything I'm going to show you here is in collaboration with Christopher Adams, who's at the Mayo Clinic. He recently moved from the University of Iowa, and this mouse work was really spearheaded by two scientists in his lab, a grad student, Matthew Miller, and a senior scientist, Scott Ebert. And what the Adams lab has shown very nicely over the past many years is that ATF4, activating transcription factor 4, is really a key promoter of age-related skeletal muscle atrophy. So what he's shown over the years is that ATF4 is required for age-related muscular atrophy. Overexpression in certain muscle types, in a few skeletal muscle types, ATF4 overexpression is sufficient to cause muscle atrophy. And together with us, he's identified using some interactome studies, he's identified some of the downstream interactors of ATF4 that are also essential for age-related muscle atrophy. But then the question is, okay, well, what's downstream of that, right? And so now we've started another study in looking at the quadriceps in these ATF4 knockout mice. And so I just want to show some data here that now what they've seen in the quadriceps recapitulates what they've seen in other muscle types, which is that in ATF4 knockout mice, in the young animals, you don't see any difference from wild type animals. But as they age, you can see that they have preserved quadricep fiber diameter. And then, so this is really an age-related phenotype, right? So there's no difference in young mice, but this difference only emerges in the aged animal. And if we look at more functional measures, we see the same thing. So again, in young animals, there's no difference in the specific force. This is like the maximum force induced in the muscle. There's no difference in this in the young animals, but this is highly preserved in the ATF4 knockouts. And in two different functional measures, grip strength and running distance, we see the same thing. Mice lacking ATF4 have preserved grip strength and running distance. So this truly is, ATF4 really is an important driver of age-related changes in the muscle. And so, so of course, we were interested in getting at this question of what's going on downstream here. And one of our first hints to look at, one of the reasons we want to look at protein turnover is that we did an RNA-seq analysis on these mice as well. And while not a lot of things changed in the RNA-seq data, the things that did change were involved in protein, seem to be involved in protein translation. So here, all of these proteins you see are CARs, Rs, Mars, Yars. These are all involved in tRNA amino acylation, important for the translation of proteins. And then we see these important translation initiation factors as well. So in light of all of these things that we found in the RNA-seq data, we decided this would be a good time to test our new tool. So we did the study using old mice that were either ATF4 knockout or control mice. We did the analysis with our turnover tool using basically the same, the same basic metabolic labeling scheme as I showed you before. And what we found was actually sort of on the bulk level was sort of unremarkable. So in these old mice with ATF4 knockout mice with preserved muscle mass, on the global level, we don't see much difference in the half-lives of proteins. In fact, there's, globally, there's no change. And if we look at a correlation between wild-type mice and ATF4 knockout mice, we can see that the half-lives are largely correlated with a few specific outliers, right? So I mean, this is particularly interesting, and I think this is a good example of why it's useful to do proteomics too, because you don't only get bulk measurements, you can look at these individual points and see what's changing. So that's exactly what we did. We looked at these individual points, and what we saw is that the majority of significant changes in the muscle were involved in mitochondrial function, or were mitochondrial proteins, or myofibril proteins. So these are core proteins in the muscle fiber. So, our working hypothesis is that changes in either of these components is really important for in regulating muscle atrophy. So, you know, and I'm open to hearing other people's suggestions on this too, actually. But what's interesting is that we took this data, so this small number of changes that are regulated by ATF4. We took this small set of changes and compared it to changes that happen during aging. So back when I was doing my PhD at UW, we had a project in collaboration with David Marcinick where we actually looked at what are the age-related changes in protein half-life in the soleus and in the EDL muscles. And out of all of these changes, the only one that showed age-related increase was IDH2. So it's really interesting. So I think one of the proteins we're really interested in now is IDH2 because this is the only protein that has an age-related change in the muscle that's actually reversed by ATF4. So this is the age-dependent change that it could be reversing. So basically, our working hypothesis now is that ATF4 is in fact very important for regulating atrophy in the quadriceps. And we hypothesize that downstream of this, that there are changes in turnover that are responsible for atrophy. And these changes are likely either altered turnover of mitochondrial proteins with particular interest in IDH2 or myofibrillar proteins. So, and these are because these are really core components of the skeletal muscle, right? So you need to maintain these to maintain your muscle mass and your strength. So that's our working hypothesis. And what we're hoping now is that with this new tool, this will be another example of where focusing on protein turnover points us to a mechanism of how we see improvement in an aging phenotype. Okay. And so now I'd like to switch gears and talk about what's been really the focus of my work during my postdoc for the last 5 years, which is cellular senescence. So cellular senescence is it's a basic aging process, and it's a complex stress response. So if you think about a healthy dividing cell, when it encounters a stress, there's a few different things that can happen. You know, first, it could be that it encounters the stress and then it recovers and continues dividing. Or it could be that, you know, the stress is too great and the cell dies, right? But there's a third thing that can happen, and that's cellular senescence. So a healthy dividing cell can encounter stress, enter senescence, and this results in a permanent and irreversible arrest of cell growth. So the cell stops dividing permanently, and then it begins to secrete a variety of biomolecules and proteins, both soluble and within exosomes, that are collectively known as the senescence-associated secretory phenotype, or SASP for short. So I want you to remember SASP because I'm going to be saying that word a lot, but SASP is this stuff secreted by the senescent cells. So with age, senescent cells accumulate in virtually all of our tissues, and via the SASP are thought to lead to aging and age-related diseases. And it's also important to know that I mentioned this is a stress response, that various different stressors can drive cells into senescence. And this includes, but is not limited to, things like genotoxic stress, like radiation, oncogenic stresses such as RAS overexpression, or even treatment stresses. So certain treatments people take in the clinic now, like the cancer chemotherapy doxorubicin or the HIV protease inhibitor atazanavir, can drive cells into senescence. So it's clinically relevant to understand, senescence biology and what's in the SASP, not only for the role in aging, but also clinically relevant for patients taking these therapies, because it's now, starting to emerge that senescent cells might be associated with some of the side effects of taking these drugs. And at this point, it's been really, really exhaustively shown that if you, using mouse models at least, that if you selectively eliminate senescent cells, that you promote health and longevity and you mitigate many diseases of aging. So when I first was going into this, I was really interested in what's going on in the SASP. And the reason for this is because it appears to be the important driver of many aging phenotypes. And we didn't really have a good comprehensive idea of what was in the SASP in the first place. So I thought, okay, let's do an unbiased analysis, proteomic analysis of what's in the SASP, because we want to know what's there in an unbiased way. but also we want to understand how this, what is the heterogeneity of this phenotype? So we know that there's different subtypes of senescent cells. For example, senescence induced by different inducers or in different cell types. So how does the phenotype vary? How heterogeneous is it in all these subtypes of senescent cells? I'm really interested in this idea that senescence, the SAS, can be used as biomarkers of senescence and aging. So right now, there's a really big push both in academia and industry to develop drugs called senolytics, which selectively eliminate senescent cells, or xenomorphics, which are drugs that alter the SAS phenotype of senescent cells as potential therapies for use in humans, right? Because we need tools, we need drugs to be able to actually apply these interventions in humans. And a really important part of developing these drugs in humans is biomarkers, because we need a biomarker to tell us, to identify a person who has an elevated senescent cell burden, and then who we can treat with the senolytic. And then after you treat them, you need a biomarker to tell you if the treatment worked. So it's really important to have biomarkers in order to translate these into the clinic. And finally, just knowing what's in the SAS will tell you about aging mechanisms. So to do this study, I used primary human fibroblasts in culture. And what I did is I induced senescence with 3 different inducers: genotoxic stress, oncogenic stress, and treatment stress, allow the cells to become senescent, and then collect the conditioned media, which contains the secreted proteins. And so then we isolate out the secreted proteins. And in this case, I'm just going to focus on the soluble fraction here. We process those for mass spec analysis. We do mass spec analysis using a really, what I think is a really cool method called data independent acquisition. And this is it's a good, cool method because it's a very comprehensive and quantitative method of doing proteomic analysis. We do the protein identification in quant, and then various downstream bioinformatics. And essentially what I found is that the SAS was much, much larger than we previously thought. So, before it was a few dozen proteins, but now we've identified, over 1,000 unique SAS factors. And more importantly, what we saw is that it's a very heterogeneous phenotype. So here I'm showing you the SAS profile of 3 different types of three different SAS profiles: genotoxic, stress-induced SAS, oncogene-induced, and treatment-induced. And what you can appreciate is that these are largely distinct profiles, right? And so this is really important because it suggests that depending on the type of senescence, that you could be driving distinct biology. And so in the interest of finding biomarkers, I wanted to focus on what are the things that are secreted regardless of the senescence inducer you use. And there were about 150 proteins here, and I call those the core SAS. And to investigate whether there's underlying biological signature to the SAS, I did a pathway network analysis. And what I found was that at least in fibroblasts, there are 3 broad components to the core SAS. The largest and most significant one by far were these pathways involved in tissue structure and organization. So most of these pathways are either ECM pathways, ECM building, or ECM breaking down, like these peptidases. We also saw this growth and metabolism arm, and this is perhaps not surprising because these are growth arrested cells, and they're known to secrete growth factors. And then interestingly, we see this neurodegeneration related arm. I find this really interesting. This isn't something we followed up on yet, but I think this is really interesting, in light of a lot of papers coming out recently showing that senescence is involved in neurodegeneration. So we have a core kind of underlying biological signature to senescent cells, but we also wanted to know, okay, which of the core SAS might be the best candidates for aging biomarkers? And to crystallize those out, we did some unbiased clustering analysis. And this really highlighted a few very key proteins in the SAS, including CXCL1, MMP1, staniolcalcin-1. So I think this is a really interesting one because, this was the first time it was ever described in the SAS. And it happens to be like one of the most robustly secreted proteins in the SAS. So if anyone has any ideas on what STC1 might be doing, in the SAS, or what it might be doing in aging, I'm really interested in hearing the ideas, because this is a really novel one that keeps coming up as well. And then of course, GDF15, And we had this interesting cluster of serpins. So we've now crystallized some of our top candidates. And so, and just to give you an idea of how robust these changes were, here are the full changes in secretion of these proteins by senescent cells compared with non-senescent cells. And when you can appreciate is that across the board, these are very high. And what I'd like to highlight is that this is a log 2 fold change. So I mean, these are like 2 to the 6th plus changes on some of these fold changes. So these are quite big changes in secretion of these proteins. And so now that we had our top candidates and we had our core SASP, we were really interested in figuring out whether these would make good biomarkers of aging in humans. And so to do this, we teamed up with Luigi Ferrucci and Toshiko Tanaka. So they're at the NIA, and Luigi Ferrucci is the scientific director at the NIA. He's also the head of the Baltimore Longitudinal Study of Aging. And what they're really interested in doing, they've been very interested now for years, is identifying protein biomarkers of aging in human plasma. And at the time, they had a candidate set of about 220 biomarkers of aging in human plasma. And we wanted to know, so we compared notes with Luigi and Tosh, and what we found is that in the core SASP, about a quarter of the entire core SASP are also significant biomarkers of aging in human plasma. And I thought this was quite a remarkable overlap, that we can see a quarter of the SASP going up with aging in humans. And some of our top candidates were some of their top candidates, so including GDF15, STC1, the serpins, and MMP1. And when I say we, I mean myself and Birgit Schilling. So this is Birgit, she's my mentor at the Buck Institute. And just to highlight one of these proteins, GDF15, in Luigi's study and Tasha's study, this protein was a single protein with the strongest correlation with aging in human plasma. And she was actually able to go back to a subset of this BLSA cohort and do ELISAs and reproduce this correlation. So I think GDF15 is another, quite a promising biomarker. And so we were really excited when we found all these interesting correlations with aging in human plasma. So one of the things we want to do going forward with Luigi is to now verify this in different human cohorts, because we don't want to, we want to see if these are actually biomarkers, these are robust across different aging cohorts, not just the BLSA, but what if we look at a geographically and genetically distinct human cohort, do we see the same thing? And so this is one of the things we looked at in a more recent study that's now in eLife. And we looked for the SASP, changes in the SASP in human plasma in the in-Chianti cohort. So this in-Chianti cohort is a representative population-based study of aging in older people living in the Chianti geographic area. And this is in Tuscany, Italy. So this is, geographically and genetically different from the BLSA. And what we found is that there is a panel of the SASP, of the core SASP, that were good predictors of mortality, and another panel, a similar panel, that were also good predictors of multi-morbidity in this completely different cohort. So, I mean, we're just really excited about this idea that the SASP can be seen in the blood, and it can also be predictive of different age-related, of aging and maybe of aging-related morbidities. Okay. So, everything I've shown you up until now has been on the core SASP, but I think one of the important things that we've learned so far is that senescent cells are very heterogeneous, and we don't know which kind of senescent cells are actually accumulating in vivo. And I think it's becoming increasingly clear that we're going to need biomarkers for specific subtypes of senescent cells. So one of the things we're now trying to do is also come up with these signatures for inducer-specific signatures of senescence. So I just want to show you an example of how we're doing this with RAS-induced senescent cells. And here you can see that in the SASP of RAS-induced senescent cells, there's a huge number of proteins that are exclusively secreted by just the RAS-induced senescent cells, but not by the other inducers, right? And this is actually not really an easy number of proteins to work with as an assay. So in order to crystallize out, the most robust signature, we actually did some multi-omic analysis here. So what we decided to do is we took this large list of proteins that are exclusive to the RAS secretome, and then we compared that to transcriptional profiles of RAS-induced senescence that were already available in the literature. And there were actually 2 studies that compared the transcriptional signatures of different types of senescent cells. And so we were able to pull out the RAS genes that were exclusive to the RAS-induced senescence from these studies, and then look at the overlap between that and the secretome changes. And we were left with 26 genes total, which is a lot more manageable. And when we looked at these, 5 of these looked like really good, robust, they would make a really good, robust signature of RAS-induced senescence. So here I'm showing the log2 fold changes of these genes in senescent cells compared with non-senescent cells. And you can appreciate that in our secretome data set and 2 different RNA-seq data sets across all these proteins, we see robust changes. So we think this is this is a really a potential approach to identify candidate panels for as biomarkers for RAS-induced senescence. So in summary, what I've shown you so far is that we've identified over 1,000 unique SAS factors. And then more importantly, that the SAS is a very heterogeneous phenotype and depends on cell type and it depends on inducer. So what we've done now is taken these different SAS profiles and we've added them to a free online database called SAS Atlas. And our hope is that this serves as a kind of a resource for researchers who think that senescence might be involved in what you're studying. You can go to this database and query, one of your molecules of interest and see if it's secreted by senescent cells. And maybe that'll tell you whether senescent cells are involved in your phenotype. And finally, I think that both the core and inducer specific SASPs are going to be promising sources of biomarkers of senescence in aging in humans. And in line with our efforts to make this data available and to make it reproducible for the community, we've also put out a few methods papers. So if anyone out there is really interested in doing, not only assessing senescence burden in vitro or in vivo, but doing these proteomic experiments, we've recently put out some really comprehensive protocols to do these analysis. So I especially recommend this current protocols paper. Francesco has done a great job of writing up all of our protocols that we used in the SAS Atlas paper, and also ways to verify that your cells were senescent. And there's another paper more recently came out in Nature Protocols, which gives a lot more, gives you more options and different assays to measure senescence burden, both in vitro and in vivo. So, I don't know if you guys get the recording afterwards, but if you do, you can, see these papers for yourselves. So now I'd like to shift gears away from the secretomes of senescent cells over to the surfaceome of senescent cells, because this is really what I want to do in my lab going forward. And what I mean by the surfaceome is the cell surface proteome of senescent cells. So this is the proteins on the surface of the senescent cells. And I think going forward in my own lab, this is something I'd really like to go to focus on. And I think focusing on the surfaceome has a lot of advantages, including it'll allow us to begin to classify the heterogeneity of senescent cells. So, just like immunophenotyping is very valuable for classifying immune cells, I think we're going to need a system to classify these very heterogeneous senescent cells. So I think it'll be really useful just for that. But of course, also cell surface proteins are potential therapeutic targets for drugs and immunotherapies. And something I'm really excited about is this idea that you can use a cell surface protein then to isolate a senescent cell from a tissue. Because everything I've shown you up until this point, the senescent cells are actually created by taking healthy cells and then culturing them and then inducing senescence in vitro artificially. But wouldn't it be a lot more clinically and biologically relevant if we could now go into a tissue that contains senescent cells and then take the senescent cells out of that tissue and study that in isolation? And I think that's a lot more, more precise way to study senescent cells, and it's a lot more biologically relevant. So this is something, going forward in my own lab that I'd like to do, and I've identified, kind of a set of benchmarks and a pipeline to get to this goal eventually. And so this entire workflow, this entire proposal starts. This is something that was originally funded by my K99 and something I hope to do in my own lab now. And this all starts by characterizing the surfaceome of senescent cells in culture. And so we've thus far been able to establish this surfaceome pipeline. And we use a well-established technique now called cell surface capture to do this. So this is a really cool proteomic technique where you use this non-cell permeable biotinylation reagent called sulfo NHS biotin. And the way it works is that you take this reagent, you add it to the live cells in culture. It biotinylates the surface lysines that are accessible on those live cells. And like I said, it's not permeable, so it doesn't get inside the cells and biotinylate intracellular proteins. So you biotinylate the surfaceome, the surface of the cells. You can then quench the reaction and process the cells as you normally do into peptides. And now at the peptide level, you can use avidin enrichment to pull down the peptides that were biotinylated on the surface of the proteins earlier on, and then analyze those for mass spec. Analyze those with mass spec, excuse me. And as proof of principle, I've done this in primary human lung fibroblasts, and I've been able to identify, hundreds of proteins on the surface of these cells, and about 100 thus far that are changing on senescent cells compared with non-senescent cells. So the next steps in this pipeline are now to validate these proteins. So I've been able to validate 3 with flow cytometry so far. And here's an example of one of these proteins, calreticulin, which is seems to be externalized to the surface of senescent cells. And what's interesting here is that even in these conditions, in these cell culture conditions, we can still see heterogeneity in the senescent cells. So only about 25 to 30% of senescent cells in culture even express calreticulin. So I think this heterogeneity issue is a really big, really going to be an important one to tackle going forward in the field. And keep in mind that this is genetically identical cells that were induced with the same dose of irradiation at the same time, and yet there's still heterogeneity emerging. And so this is really a reoccurring theme with both in my data and others who do single cell sequencing. So I think this is going to be a really important issue in the field going forward. So after we validate these markers in vitro, we want to validate them in vivo. So the idea here is now working together with Luigi Ferrucci, we'll obtain specimens that contain senescent cells, and we'll validate whether these surface markers are on those senescent cells. And if they are, then we consider those validated, and then we can do the fun stuff, which is to try to isolate these cells from human tissues. So idea here again are in collaboration with Luigi Ferrucci. We get punch biopsies containing senescent cells. We make single cell suspensions and then use that surface marker to then sort out the senescent cells from those suspensions. And now we get to do the cool stuff, which is molecular phenotyping of isolated senescent cells from in vivo for the first time. And again, I think this is really a lot more biologically relevant to the kind of senescence that we care about when we study senescent cells. So this is the end goal. And I just want to end by saying that I think this helps to address some really important challenges in the senescence field. And I think, some of the really important things we need to address is that we need tools to quantify and target in vivo senescence, whether that be with biomarkers or these cell surface proteins. But we also need to understand the heterogeneity of the senescent cells, right? And we really need to get at this question of how do phenotypes relate to aging phenotypes. And by phenotypes, this is a word I made up, but by phenotype, it basically means the repertoire of senescent cells in an organism or a tissue, right? How does that repertoire of senescent cells affect the aging phenotype? And I think this is going to be really important to understand and try to figure out, because as we now know, senescent cells are really heterogeneous. Their phenotype depends on the inducer or their cell type. In vivo, senescent cells have different functions. We know that some senescent cells, are accumulating and causing aging, but on the other hand, there are senescent cells that are helping us in wound healing. And we're going to need to be able to distinguish one from the other and target one, or target and quantify one versus the other. So these are really important issues in the field going forward. And, and we also see this heterogeneity arise even in culture. And so these are some issues that I think we can really address by focusing on the surfaceome, because again, we can use the surfaceome to classify the senescent cells, target them potentially with drugs or immunotherapies, or isolate them. from clinical specimens, and this would be a lot more clinically relevant way to look at senescent cells. And this is exactly these are exactly the kind of issues that I hope to address in my own lab going forward. And I just wanted to plug my lab because I'm going to be starting a lab at the NIA in July of this year. So if there are anybody who's looking for a postdoc or RA position, the Translational Geroproteomics unit is who's gonna be interested in addressing these kind of questions, and some of those outstanding questions and turnover I mentioned earlier will be some of the things we hope to address in the lab. So if you're going to be looking for a position later this year, feel free to reach out to me or get in contact with me now. My email address is down here. And this is going to be at the NIA, which is in Baltimore. And with that, I'd like to thank all the people involved in this work, which were a lot. So for the turnover work, the Schilling lab, the Makas lab, and the Rabinovich labs for all the collaboration and help on this. The senescence work, again, with Schilling lab and Campisi labs, the Verdun lab, and then of course my collaborators at the NIA, the Ferrucci lab, and on the surfaceome work, Brian Woolshide and Jim Wells, and of course my funding sources. And thank you all for your attention, and I'm happy to take questions. Thank you.
Great job, Nate. It's really nice to see how your work has continued, but really grown since the time that you were here in Seattle. And also, it's nice to see that your collaborations, relationships remain strong too with our local community. So thank you for that.
Oh, yeah.
I invite everyone to either put their comments and questions in the chat or raise your hand and I'll call on you. There's one from a current first-year student in our program asking, what would the methods be to increase protein half-life that you mentioned in the first part of your talk? And would those affect the surfaceome? Does it control control cycling of the cell surface markers?
Oh, that's a really good question. So I think at your first point, yeah, I think a lot of the aging interventions we already use and know of seem to increase protein half-lives globally. So I think there are a lot of interventions out there already that we can expect will increase protein half-lives. But getting at your second question, that's a really good question. And that would be really interesting to look at. So we've never focused solely on the surfaceome, but I wouldn't be surprised if there were interventions that increased half-life of the surfaceome as well. But what we do know about the surfaceome is that it's extremely dynamic and it responds very fast to stimulus. So it's one of the kind of quicker responding groups of proteins, I guess you would say. And so, when we look at the surfaceome, we also have to be careful about that.
I guess I have a follow along to that then. As you isolate these cells, either in vivo or in culture, if it turns over quickly or changes quickly, putting the cell in culture itself is a kind of an injury response for the cells. And so how confident are you that the things that you observe in a cultured cell are exactly what you observe in vivo?
Yeah. No, that's a really good question. That's the thing that actually worries me the most about doing this. And I think that's why one of the really important, so one thing that gives me kind of reassures me a bit is that we've been able to, of the markers we found so far, calreticulin, we've at least been able to verify in mice in vivo. So that's good, but that is definitely a worry. I think this is kind of a funnel approach where you identify a lot of candidates, and you validate those in vitro, but then I think my expectation is that a lot of them may be lost when you get to the in vivo stage, and that's something yet to be seen. One thing that I am kind of reassured by is that one way that we've been able to identify kind of robust secreted markers is by looking at the core SASP. So maybe we can kind of focus on the core surfaceome as well. I think it's really amazing that cells, cultured senescent cells, and what they secrete is actually reflected in humans too. So maybe, that kind of reassures me that maybe we could see the same thing with the surfaceome of senescent cells.
Great, thank you. Daniel Promiso asks, in thinking about your collaboration with Luigi, are you interested in multi-omic approaches, combining proteomics with other domains? And if so, what do you see as the challenges and potential benefits of this approach?
Oh, sure. Oh, that's a good question. Yeah, definitely. I think as we've already seen, with some approaches like, epigenetic clocks and things, that these approaches all already work really well to quantify aging. I think it does have there's potentially benefit, I think, in using multiomics. I think adding more information into modeling senescence burden outside of just the proteins in the plasma will be really useful. And I think especially in expanding the scope of the proteomics may be really useful. So, because I think one thing we really see with all of these kind of aging clocks in the plasma proteome is that they're pretty good, but not super good. And I think part of the reason for that is that, we're not catching all the dimensions of the proteome when we look at these with traditional approaches. Like when you use like a SomaScan approach, you're really not capturing any information about the proteoforms, but maybe using mass spectrometry, you can actually capture some information about the PTMs and things in there. And maybe, because we're not capturing exactly which proteoforms are there, that's what's causing some of the noise in these models, right? That aren't super well predictive of aging. Maybe we can make it a little more precise by layering on information about the, which proteoforms we're looking at, or if we can layer turnover information in there somehow, that would be really, I think, potentially useful. I hope that answers your question, Daniel.
He's shaking his head, yes.
Thanks.
I see a couple of people have turned on their cameras. If that's indicative that you want to ask a question directly, I welcome you to do so. Or if you're just there to smile, then that's fine too. Smile is for one. And Mike?
I'm glad I don't have a specific question. I do more of a comment. I'm always kind of concerned whenever I see SomaScan and what people are actually measuring. So it'd be good to see other methods being applied. I think it'd be good.
I agree.
Yeah.
And I mean, yeah, and with these really big, I guess one of the upshots to that is that they're supposedly so comprehensive, but it's, I'm always a little skeptical of how, I guess
measuring lots of things poorly is not
yeah, exactly.
It's not giving me more confidence.
How specific are they is the question. So I think, if we use more specific methods to measure some of these proteins, that'll give us more confidence in them too. Yeah.
That's great.
Yeah.
Well, there's one question and I have one other as well. So do differences in mass isotopolog distributions infer anything about the proteome among the labeled peptides?
Differences in mass isotope. Yeah. So I guess, so you mean on the level of individual peptides or proteins? So it might infer something about the proteome. So I guess what we use the mass isotopolog distributions for is to actually help us calculate the enrichment in the precursor amino acid pool. And when you see a wide distribution of from all of your different peptide measurements, if you see a really wide distribution of isotopologs, or different distributions among different peptides, then you might infer from that there's not a single compartment of amino acids in the cell or something like that all the proteins all the peptides are drawing from or being synthesized from. Beyond that, it's hard to say. I think one thing that I don't have the slide available now, but in one of the things we do look at in these studies is we look at we take basically the mean precursor pool calculated from the median of all of our different peptide measurements and their mass isotoper distributions or isotopolog distributions, and we see that they tend to peak at around one value, but I'm not sure if that's the case in all studies. I think there are other people who use multi-compartment modeling and things like that, so it could be something else. There could be a different case as well.
Wonderful. Okay, we'll take one last question here. I'm wondering if the tRNA amino acylation complex protein data and whether these fit with Ed's finding of redox regulation of Mars and Lars. And I'll let you interpret that question because it's outside of my realm of expertise.
Oh, that's a great question, Devin. Oh, thank you, by the way. That's, that's a great question. I'm not familiar with those studies. This is actually really fresh data for us. So I'd be really interested in actually getting those studies. If you don't mind sending them to me afterwards, this might be really interesting in the context of the muscle atrophy study. So, okay. Yeah, sorry, I don't know because I'm not familiar with those studies though.
All right. So with that, looking at the time, I want to thank you all for joining us. And I want to thank Nate for coming back and showing us what a successful trainee looks like and all the great things that you've done so far. And we have great hopes for you going to Baltimore and NIA, hopefully after the cicadas have come out and done their thing, and you can then enjoy hot and steamy Baltimore. I put a note in the chat for our trainees. These are undergrads, grads, and postdocs. We're going to have a separate Zoom conversation with Nate, so you can kind of ask some more things about lessons learned and how he got to make the decisions in his career. It's a great opportunity to learn from our own alumni. So please do join us in that other Zoom room. I'm going to wait here till this one closes, then I'll go and open that one up. So we'll probably start at about 2:45 to give Nate a minute to catch his breath before we start up. But, thank you all for joining us, and we'll see you at the next Path Presents. And thanks again, Nate.
Thank you.